Hello, this is Tommy. Welcome back to Chatomics. I have over 10 years experience in computational biology, and today I'm going to explain what is a Seurat object for single-cell RNA sequencing data analysis. And I will also demonstrate a real example in RStudio. Okay, so first of all, what even is an object? So Seurat is a package for single-cell RNA sequencing data analysis, and it contains core data infrastructure called Seurat object. So what even is an object? You can think our whole body is an object, right? Then you have clothes, you have pants, and then in the pants and in your clothes you have pockets, right? And in the pockets you can contain different objects as well. So you can have keys and you can have multiple keys, right? So in Seurat, a Seurat object, for example, in this case has 10 slots. So this is in our terms, like slots is more like a pocket, for example, in your body. So it has, for example, at least in the old version of Seurat, I will explain actually the later V5 version, but in the old version, for example, a Seurat object has 10 slots. Okay, it has 10 pockets. So it has the assays and the metadata and reductions and other stuff here. And in R, how you access a slot, so you can use this @ sign. For example, if you want to access the assays, so you use the object name PBMC @assays. The metadata is just a data frame for that metadata slot, so you can access that by @ sign as well. Okay, and for each actual slot, it can also contain different objects, just like in each pocket you can contain multiple keys or phones, different objects, right? For example, in the assay slots, then you contain the assay object. If it's an object, it can also have many different slots, right? So an assay objects contain, for example, 6 slots here. So you have the counts, which contains the raw counts and the data, the normalized data, scale data that is actually z-scored. Okay, and for the reduction slots, it contains, of course, the dimension reduction objects, and the dimension reduction object contains a couple of slots as well. For example, the cell embeddings, the feature loadings, okay, and many other stuff. Okay. So the key to really understand the Seurat objects is to understand how to access all those objects within the slots. Okay. For example, the dimension slot of the Seurat contains a list of the dimension reduction objects. So remember, you can have the PCA, principal component analysis, UMAP, or t-SNE, those like non-linear dimension reductions. So it's a list, okay? The reduction slot contains a list of objects. So if it's a list, for example, you can use the @ sign to access that slot, and then use the $sign to access each element of that list, right? So either PCA, UMAP, or t-SNE. Okay, this is just a reminder how it looks. And so remember, you can, for example, here, the reduction objects has different slots, so you can access those slots using the @ sign as well. So in this case, the PBMC is the Seurat object. You access the reductions slots, and it has a list of dimension reduction objects. So let's use the $sign to access the PCA objects. And for that PCA object, it also contains multiple different slots. One of them is called cell embeddings. Right? If you use @ sign here, then it will give you that principal component for loading embeddings for each of the cells. So each row is one cell, each column is a principal component. Okay, and this way to access the objects within the slots is kind of too long to type. So Seurat has this operator overloading for easy access of those objects. For example, you can use PBMC, so double brackets, and PCA to access that PCA object. But of course, you can use this long way. So at reductions, that slot contains a list of objects and $PCA, right? Okay. So, and it also has other getter functions to easily get those data. For example, you can use embeddings or loadings and to access the gene loading or the cell loadings for the PCA easily, a little bit easily. Okay, let's actually get into RStudio and showcase how do we do that in real time. Okay, to do this, let's actually load the library, Seurat, and library deploy. And you can check your Seurat object, Seurat version, the package version. So now I'm using version 5.1. So Seurat version 5.1 actually implement a new assay object v5, and it has other advantages. For example, it can contain the matrices on disk, so you can actually deal with bigger, much bigger data sets. But the concept is very similar. Like as long as you understand the slots, and you can easily actually access all those objects within the slots as well. Okay, let's load the Seurat data package and get the PBMC 3K data set. Okay, so it's pre-built in this package. So if you look at this, you will say error because I think it's an old version of Seurat. So you can use the update Seurat object to update into the most recent version. Okay, so it's updating that, and now if you rerun the PBMC, now it prints out the information without any errors. Okay, and then you will see here actually now you have actually the layers instead of the slots actually. So, but it's essentially they're very similar. Okay, you will see. So let's actually just do a very routine pre-processing for this object. So normalize the data, find the variable features, scale data, and then run PCA and the UMAP for dimension reduction. Okay, so it should be very fast. Okay. And as you can see here, Seurat v5 assay store data in layers. And previously, those are called slots. Okay, so there will be raw counts, the normalized data, and or the z-score data like scale.data. Okay, so you can use the layers function to look at. Okay, now you have these 3 different layers. But in old Seurat version, those are old called slots. Okay, so you can actually access the counts by using the layer data function and counts, and look at the first 5 rows and 5 columns here. And also you can look at the normalized data in a similar way. So this is the old way for Seurat v3 or v4. So you use the get assay data, then you slot, right, instead of using layers. But those functions give you essentially the same results. Okay, it tells you, okay, this is the same thing as actually this one. Okay, so as I mentioned, so PBMC3K, this is a Seurat object, so you can access different slots using the @ sign. For example, the metadata is one of the slots. So you use this and look at the first 6 rows. It's just a data frame. Okay, so each row is 1 cell, each column will be 1 features for that cell. Okay, and again, we can use the @ sign to access the assay slot. And with this, you see here, it will return you a list of assay, right? Assay data. So one of them will be RNA. So you can use the $, it's a list. So use the $RNA to access that assay data. And of course, you can use the shortcut because this double bracket operator, then essentially get the same thing. Okay, and you can, for example, you get the assay, the RNA assay here, assay data. It's an object, so that object also has slots. Okay, and then you can access the slots using the @ sign. Okay, if you use this, it'll give you the full counts matrix. But if you do that, it'll give you the first 5 rows and first 5 columns. Okay, similarly, you can look at the normalized data. Okay, now let's get into the dimension reduction. Okay, so dimension reduction. So those objects, dimension reduction objects, they are in this reduction slot. Okay, so you use the @ sign to access the slot, and it will tell you this is a list of dimension reduction objects, right? So now you have 2 PCA and the EOMAP, and then you can use the $sign because of the list. You use the $sign to access the elements. You use $sign PCA, so it gives you the dimension reduction object. And that object has different slots as well. One of them will be the cell embeddings, right? So you can use the @ sign again. So remember, always access the slot, use the @ sign. Okay, so if you use @ sign to access cell embeddings, look at the first 6 rows here. Then each row will be 1 gene, each column will be 1 principal component. Of course, as I showed in the slides, you can use the double bracket operator to access the dimension reduction object as well. So this is a shortcut of this. Okay, and then of course you can access the cell embeddings. Use the @ sign because this is the PCA object. @ sign to access that slot. Look at the first 6 rows. It gives you the same results. Of course, Seurat has other useful, easy to use getter functions. For example, you can use embeddings, the object name and the PCA, and then look at the first 6 rows. It gives you actually the same results like this. Okay. Of course, you can also look at the embeddings for EOMAP, and you can also look at the feature loadings or the gene loadings from the PCA components, principal component analysis. Okay. So you can also convert the different assay types from V4 and V5 and interchangeable and vice versa. So remember, let's make a copy, PDMC, and you can convert the V5 assay to V4 assay using the as function and define the class to assay. And then if you look at the class again, it's called assay, but you can convert it back, use the as function, but you define the class to be assay5. And then if you look at the class again, it will be assay5. Okay? So as long as you understand the structure of that Seurat objects, what kind of slots are there, and how you access the slots using the @ sign, and you understand in each slot it contains a list of objects or maybe just a single data frame, you can easily actually use the @ sign and $sign to access all those elements of the Seurat objects. So I hope this video is helpful for you, and make sure you subscribe to the video, and happy learning, and see you next time.